All right. Hi, everybody. Thank you so much for coming today. So last time we had kind of left off with going over the heart and the circulation of blood flow through the heart. So today we are going to finish that up with the electrical signaling through the heart. So the electrical signaling through the heart is going to be tied to the circulation through the heart. So basically, in order to understand electrical signaling, you kind of need to have a basic understanding of flow and then vice versa. Oh, we're already leaving our breakout rooms. Okay, well, that worked out. I'll see you guys out there.
Because everyone
Recording
else is
in progress.
done for the day, so don't worry about it.
Dan, are you talking? No.
No, I was Amy was having trouble. There's I had someone get like just kind of boomeranged out of breakout rooms this week. And it's happening to Amy as well.
That would
so
just happen to us?
No, you guys are fine.
Oh, okay, we're good. Okay.
Yeah. Carry on.
Okay, sounds good. So basically, when we look at the electrical conduction through the heart, we're going to start with a few like general concepts and then get into more details. So remember that the heart is autorhythmic. This means that it has its own rhythm. So it's going to keep conducting itself without necessarily needing input from the brain. So what this means is the heart beats on its own without needing stimulation. So you can still speed up or slow down your heart rate. Your body can still do that. But if you took the heart out of your body and you put it on a table without any stimulus from the brain, it would keep beating. So the systole of the heart is the contraction of the ventricles. So the contraction of the heart when it's pumping, and then diastole is the relaxation. I love you. And then we're going to start talking about how that actually happens when we look at the electrical signal. So the heart, right, has our 4 chambers we talked about last time. Right atria, right ventricle, left atria, and left ventricle. And what we're going to see is there are different nodes that go throughout those chambers. So we start with the SA node. So that's going to be number 1. SA node, atrial node, and this is going to be your first electrical signal origination. So this is the first place the electrical signal starts in the heart. So we're going to start over here, and then we're going to send that to the AV node, number 2. So AV node is the second location, and that's atrioventricular node. So you can see that that's kind of in between the atria and the ventricle, right? Kind of that spot right here. And then we're going to go to our interseptal fiber next, our bundle of His next. So right here, this is going to be number 3. Number 3 is the bundle of His. So this is the next location that the signal travels. Then it goes down interseptal fibers. So that's number 4. And then the last spot the signal travels is up the Purkinje fibers, which kind of loop all the way through the heart muscle on either side. Okay, so that's number 5. Purkinje fibers. And one Purkinje fiber will start the next signal. So this is number 5. So we start with the SA node, it sends it to the AV node, then we go to the bundle of His, then the interceptal down the middle of the heart, and then up the sides. And one Purkinje fiber, this guy right here, is going to restart the signal. The SA node is also called the pacemaker. Okay, so what we're kind of expecting you to know here is we want you to know the flow, what goes to what. So what starts it, the SA node, where does it go next, the AV, then the bundle of His, then the interceptal and Purkinje, and then kind of also what's happening, right? The pacemaker of the heart. The heart is setting a rhythm and it's going to pump out blood based upon all of this flow through the different chambers. Okay. All right, so remember when we learned about, do you guys remember learning about the myocardium, the cardiac cells last unit, and you learned about the intercalated discs. And remember how those had sodium. They allowed sodium to pass through really quickly. Well, when we get into all of this, the SA, AV, bundle of His, and interceptal, that's where we actually see those intercalated discs that you guys learned about before. So they're kind of coming back here, helping those signals to travel all throughout the heart chambers. Okay. So remember what we start with and what we finish with. And now we're going to talk about the PQRST wave, and then we're going to link it back to how that relates to the different conduction through the heart. Okay. So when we go to the PQRST, kind of looks like this. So this is the P, Q, R, S, and T. And each of them has a meaning of what's happening in the heart. So let's start with the QRS, because that's kind of the one we already know about. So when the QRS complex occurs, the ventricles contract. So remember, we drew our heart Thursday. We said that there was a right atria, right ventricle, left atria, left ventricle, right? This side pumped out to the lungs. This side pumped out to the body. So when the ventricles contract, that would be the left ventricle here, sending blood to the body, and the right ventricle sending blood to the lungs. So the right ventricle sends blood to the lungs, and the left ventricle sends blood to the body. That's what happens during the QRS complex. Okay, so this guy fills with blood here and sends it to the body. Remember, that was through the aorta, and then to the lungs. What was that through? The pulmonary trunk. So then we want to go back to the P wave. So in the P wave, the ventricles are filling up completely with blood, requiring the atria to contract to finish filling them. So what happens is the heart, try to draw it in kind of step by step. We'll say this is step number 1. So the blood is going to pass through those cuspid valves, right? Down here and down here. So this blood came from the lungs, right? This blood that enters into the atria here comes from the body. And so that's going to fill up the top chambers, right? Coming from the lungs, coming from the body, fills up those top chambers. So this is our step number 1 here. And then in step number 2, we get that blood passing down into the lower chamber. But only 80% of blood flows down into the ventricles from the atria passively. So what this means is 80% of this blood that's up here in step number 2 flows down passively, right? So we get these ventricles are 80% full of blood, but they do not have 100%. Do not have 100% of the blood that was at the top. Okay, so we fill them up, but only to 80%. So what we do in step number 3 is we're going to force this last little bit that's left. So this would be 20% up here, 20% up here. So in step number 3, the atria contract to send the last little bit of blood down. So right atria, right ventricle, left atria, left ventricle. And we said that 80% is down here, right? 20% is left up here. So in step number 3, the atria contract to send the blood down to the ventricles to fill them 100%. Okay, so this last little bit gets contracted or pushed down. And when we actually contract the atria, we call that atrial contraction here or depolarization. So what happens in the P wave is that last 20% of blood is sent to the ventricles, sent down to the ventricles. So that's the P right here. That's when the atria contract, okay? So we said that the QRS is when the ventricles contract, right? So that's because when we actually get all of that blood down, we'll just pretend this is now step number 4. Once we get all of that blood down here, 100%. So this is now step 4. Then the ventricles can send that blood to the lungs and to the body. So the QRS complex will come after the atria contract and send the last 20% down. We will then get the ventricles contracting. Okay, let's erase this guy over here. All right. And then the last step is the T wave, right? The T wave comes next. So PQRS, right? And then T. QRS, we said that that was when the ventricles contract. And last we have the T wave, okay? So during the T wave, what happens is the ventricles are refilling. So during the T wave, the ventricles refill with 80%. So remember when I talked about that part where they refill with the 80% passively, requiring no energy? That is what's happening in the T wave. So in the T wave, the ventricles refill with 80%. Then we're going to go back to the P wave again. Does that make sense? So in the P wave, right, the last 20% of blood is sent down to the ventricles. 80% got down here. And then when we get over to the P wave, the last 20% is sent down. And again, this would be the QRS right here when those ventricles are contracting. Okay? P wave, the atria contracts, sending the last little bit of blood. QRS, the ventricles are contracting. And then during the T wave, the ventricles refill with that 80% of blood. Okay, all right, so any questions there? All right, so what
Quick
happens?
question, I'm sorry.
No, go ahead.
So the way it works, so it starts in the P wave. No, does it start in the P wave, right?
So this is the reason it's hard. I'm trying to like slowly explain it. I know that it's hard. It's kind of confusing. The reason it's confusing is because you really just pick anywhere you want to start, because
Okay.
there's not really a place that it starts or ends, because it's like it's always going. So it's just easiest for us to say, okay, let's start the P wave. But that's kind of why I had to like explain before we got to the P wave, because it's like, wait, where's this 20% and 80% thing coming in? So if we start at the P wave, let's just choose the P wave. We'll say, okay, let's start there. The P wave happens when the atria send the last 20% of blood down to the ventricles, because the ventricles could not fill with that last 20%. It's almost like they're too full. And then as soon as they get full, they contract, which is the QRS complex. They get so full, they contract. And then once they're done contracting, they fill back up again, which is the T wave, when they fill back up with that 80%. And then that brings us back to the P wave again, because now there's 20% at the top, 80% at the bottom, and we have to go back over to the P wave and send it down again. Does that make sense?
Yeah.
It's kind of like a revolving door. Like you just choose one to start with. We usually start with the P, and then we say, okay, that's then the QRS, and then the T is when they actually refill down here.
Okay.
So atria contract, the top ones contract in P versus the bottom ones refill in T.
Okay. Thank you.
Yeah. All right. So there's only one thing we're missing here. And I just wanted to make sure hopefully everybody feels like it's kind of something in there is making sense. The atria also have to fill back up, right? So we said that in the QRS, the ventricles contract, right down here, ventricles contract. And the atria contract in the P wave over here, right? And then the last step is that the ventricles refill on the T wave over here, right? When is it that the atria contract? The atria are going to contract while the ventricles are going to refill while the ventricles are busy contracting. So during the QRS, the atria refill. So what we're saying is that the ventricles are busy contracting and sending all of that blood to the lungs and to the body. And meanwhile, the atria are refilling with more new fresh blood coming from the lungs and the body. So the biggest thing that you have to kind of think is, okay, so your heart is never going to waste time. It wants to use all of the time effectively. So while the ventricles contract in the QRS, the atria refill. Does that make sense? They're refilling while the ventricles contract. So what we say over here for the QRS complex is that the ventricles contract and the atria refill. The word we use for refill is repolarize. The word we use for contract is depolarize. So just think of those as just synonymous. Think refill, repolarize, contract is just the other word, depolarize, okay? All right. So T wave, if the ventricles refill, that means the ventricles repolarize. Okay, we're going to apply it now to the SA, AV, and bundle of His and interseptal fibers. Any questions here? Start with the atria contracting, last 20%. We move to the ventricles contracting, the atria refilling, and then we get the ventricles refilling in T wave. Okay. So we have our P, Q, R, S. I think someone has their mic on. Okay, you have your mic on, just mute yourself. Okay, unless you have a question. So again, P, atria are going to contract. This is the last 20% of blood sent to the ventricle. Okay, and remember this would be depolarize. Atria depolarize in P. This is Q, RS, we call it the QRS complex. This is when the ventricles contract or depolarize. And meanwhile, the atria repolarize or refill. So while the ventricles are busy sending blood to the lungs and to the body, contracting, the atria are both refilling. And then last, we have our T wave. This is when the ventricles repolarize or refill. Right now in my third semester, we're learning about PQRS T waves again, but this time we're learning about how to interpret them on a patient. So like if you had a patient and they have problems with the PQRST, what does that mean about their heart, right? So the biggest thing that you guys want to walk away with is an understanding of what's going on, right? So you need to make sure that you guys get what's going on if you're planning on using this information when you go forward from here, okay? So when we look at the SA-AV bundle of His, interceptal, and Purkinje, we started with the SA, we progress to the AV, right? And then we said next one was the bundle of His here. And then we go down, these are the interceptal fibers. And then we go up the Purkinje fibers. And one goes back to the SA node. Okay, so let's figure out which nodes are sending the electrical signal during which parts of the PQRST. All right, so if the SA, which is the sinoatrial node, sends a signal to the AV node. Okay, hopefully everybody is with me what I'm doing. So I'm talking about SA sending a signal to AV. So where is this happening? What do you think the chamber is that this is happening in? It's happening in the atria. Does everybody see that? SA would be sending a signal to the AV node in the atria, okay? So SA to AV is causing atrial depolarization. If SA to AV is causing atrial depolarization, what phase of the PQRST wave, what phase of the PQRST is this? What would be SA to AV? Which phase? P, right? P. So the P wave represents the SA to the AV because that's when we have atrial depolarization. Good. Okay, so what's our next step? We have our AV conducts our signal to the bundle of His, okay, to the interceptal fibers, to the Purkinje fibers, right? Kind of moving through the ventricle. So the signal moves through the ventricles. So what do you think if the signal is moving through the ventricles, what is it making happen for them? So what is happening in the ventricles when the signal is moving through? Is it A, depolarization, or B, repolarization? Which one is happening in the ventricles when the signal moves through? So depolarization is contraction, repolarization is refilling. Good, right? So a signal moving through the ventricles, this is causing electrical activity. If it's causing electrical activity, it's causing depolarization. All right, so the AV to the bundle of His to the interseptal to the Purkinje moves through the ventricles. So what that means that we have ventricular depolarization. If we have ventricular depolarization, which phase are we in? We have ventricular depolarization. Which phase are we in? QRS, right? Good. QRS complex. So those are the two main kind of things that we look at with the SA to the AV would be atrial depolarization or the P wave. And then the AV to the bundle of His, interseptal, and Purkinje would all be causing ventricular depolarization. For repolarization occurs after the signal travels. So again, like I said, when we're talking about the SA to the AV node, we're actually saying that the signal is traveling from one to the other. After it's done traveling, repolarization will occur. So we don't see repolarization really on the SA to AV to the other ones because it's only depolarization. Okay? All right. So I know that it was a little bit, it's a little, it's kind of a lot. It's a little confusing of an area of the lecture. I would just encourage you that it's very confusing at first, and then the more you kind of go over it, the more it does start to make sense. So I would just encourage you to kind of look at it a little bit more before, again, like going to the test, you're going to want to touch on it a few more times. But that's the basics of it. I would say that this picture here is probably pretty helpful, showing you how the P wave has the atria contracting the last 20%, and then how the QRS has the ventricles contracting and the atria refilling, and then the ventricles refilling in the T wave portion. Okay, so I'm gonna move on from this topic, and we're gonna talk about the actual like blood vessels, arteries, arterioles, and capillary beds and all of that next, and then we'll get into blood itself. All right, so when we leave the heart, we're going to take, we have the heart, and we know that it starts with the left ventricle pumping out that blood through the aorta. And then if you guys have done your blood flow, we know that the aorta is going to branch first into arteries, and then into smaller vessels we call arterioles. And then those will become the capillary bed. These are arterioles. And then that will then branch into our littlest vessels called the capillaries. And then this is what we call our capillary bed. So the capillary bed is when it joins up with the venous side. So this is a capillary bed. A capillary bed is where arterial capillaries meet venous capillaries. So the blood is going to start coming back to the heart. So first, it's going to join up into a little bit bigger vessels, and those are called venules. So this matches, look over here, arterioles and venules, these would be the same size. So let's say a capillary bed is an extra small, arterioles would be small, arteries are going to be medium, and then the aorta is large. So the venules are going to join together and become veins, And those veins are going to travel back to the heart. Trying to get myself back to the heart here. And then that'll all join together as two different large vessels, right? The inferior vena cava and the superior vena cava. So those are going to be the large size here. This would be medium and small. So the way that Knudsen describes it that I really like is if you picture you're driving on a freeway and you're on the freeway, this is you, your little car on the freeway. You take the off ramp, so this is like an off ramp. Now you're on the side streets, right? Then you're on your little street where your house is, like a little tiny house street. Then you go back to the freeway. So you're headed back to the freeway. You have to get on the streets again. Then you have to get on an on-ramp and then back on the freeway. Maybe this you could picture as like the freeway going south, the freeway going north. You have to get off, get on the streets, little tiny houses or the capillary bed area, back to the streets, the on-ramp, and then the freeway again. Does that make sense? So your blood is big, and then it begins to get smaller, and then goes back to the heart getting larger. So on the venous side, the blood loses pressure. So because the blood is constantly going from this small little vessel to a bigger vessel, to an even bigger vessel when it's through the venous side, the blood is losing pressure and losing, like it's not being propelled well, not getting back to the heart well. So your body has valves. Reason for valves on venous side. So the valves on the venous side, in essence, keep blood moving back to the heart. There's no need for valves on the arterial side because that blood is flowing really nicely all the way down. But as we actually start to go back to the heart this way, we go from small little tiny vessels to really big vessels, and we lose pressure. So what your body has is it has in place these valves, and each time the heart beats, the blood is kind of, I picture it, pushed up to the next valve. Let's look at one of these valves. So here's your blood. It's making its way up, right? And your heart is busy beating, trying to pump it back up to the heart. It's going to go through this valve, and then when your heart relaxes, it wants to come back, but the valve will close and catch it and keep it from coming back, so that the next time the heart pumps, that blood gets into the next chamber area, tries to come back, but it can't because that valve closes. Once again, the heart beats, and that blood keeps moving up because the valves catch it and keep it from returning and flowing back down. So the valves, in a sense, are once again preventing backflow. So these valves prevent backflow, and so these help the blood return to the heart. Does that make sense for why the arterial side does not have valves, and then why the venous side does have valves? And then also the sizes. So for your test, you do need to know, like, arteries go to arterioles, veins go to venules, all of that. All right, so let's talk about the coverings around each of these. So if we're talking about the arteries and the veins, we remember that those are the same size, right? I'm going to go to a different page. Arteries and veins, arterioles and venules, and then the capillary bed would be the same size. Okay, so if we're talking about, again, arteries and veins, these are large in size, not as large as the aorta and vena cava, but they're large. And then the next one we have are the veins, the arterioles and venules. And then the last one is the capillary bed. Okay, so the covering around the arteries and the veins consists of all three layers, as does the medium in size arterioles and venules. There's going to be all three layers. So the layers are tunica externa. This is the outermost layer. So if we kind of picture a vessel, tunica externa, be kind of this outer, and it's dense, elastic connective tissue. Dense, elastic connective tissue. Second one is our tunica media, and this is our smooth muscle layer. So smooth muscle here. Okay. So that's number 2. And then the third layer is the tunica. Let's do green. No, too black. Tunica interna. The tunica interna is simple squamous epithelium. And this is the part that allows diffusion to occur. So this allows nutrient and gas exchange to happen. So that's going to be the innermost layer here. All right, so blood is flowing through this blood vessel, right? And it's nice and protected. It has all three layers, if it's an arteriole, a venule, an artery, or a vein. Okay, so this is true for pulmonary, systemic, and coronary circuits. So remember how we talked about the pulmonary supplies the lungs with blood? It goes from the right ventricle, pumps to the lungs, comes back to the left atria, right? Goes to the lungs from the right ventricle, comes back to the right, to the left atria, then goes down to the left ventricle, and that was the pulmonary. So those have the same vessels: pulmonary arteries, pulmonary arterioles, pulmonary capillaries, and then this was coming back as pulmonary veins, right? Remember how we said pulmonary arteries, arteries always go away from the heart. This is really important for your exam, right? Arteries always go away from the heart. Veins always come back. So if you're in the pulmonary circuit, this will be helpful for you guys when you're on your cardiovascular lab packet. In your pulmonary circuit, those will be arteries will have no oxygen going away and veins come back to the heart. If you're systemic, that goes to the body, right? Leaving from the left ventricle, going out to the body. And that's the one we talked about just now. And then coronary supplies the heart itself. So like your actual heart muscle is supplied by the coronary arteries, arterioles, and capillary bed. All right, so let's talk about the capillary bed. So whenever we get down to a capillary bed, this is where the arterial and venous capillaries meet. And in the capillary bed, we have oxygen and gas and nutrient exchange, right? That's where all of that good stuff is getting exchanged. And if you remember, number 3, the simple squamous epithelium is the only one that allows the nutrient and gas exchange to happen, right? It's the only one that allows that to occur. So if that's the case, then the tunica interna is only found in the capillary bed. What this means is there's only one layer, and that layer is the tunica interna. There's no tunica media and no tunica externa in the capillary bed. This is important because what we're going to learn about right now is we're going to learn that that tunica interna can change and open up and develop holes that allow the passage of certain things to go in and to go out of it. Okay. So if we think about a little bit of kind of a diagram of what we're doing, we talked about the PQRST and the electrical conduction through the heart. That was the first thing we talked about. Now we're talking about the kind of the anatomy of blood vessels. We talked about their basic structure, anatomy, and now we're going to talk about some function of the capillaries, and what that looks like is the different kinds. So we're going to explain continuous capillaries, fenestrated capillaries, sinusoid capillaries, through the process of diapedesis. Okay, so that's kind of what we're talking about right now with capillary function. Okay, so that was anatomy and then function of capillaries. That as soon as we're done talking about this, we're going to start talking about blood. So as we get going into blood, we're going to do some functions of blood. So the composition and anatomy of blood, which is like plasma and the formed elements, plasma and formed elements. And then we're going to talk about blood clotting. And at that point, what I'm going to do is pause, and we're probably going to go over some practice questions. And then I'll pick up again on Friday so that you guys have plenty of time to kind of catch up, because I don't know that I want to get too far ahead. We'll kind of see how it goes. How many of you have not watched any of the blood stuff? I kind of am curious to know, because I don't want to. Basically, I'd like to start it, but I don't know if I should finish it. Okay, so how many people have not? I'm fine with starting it because I think that would give you guys a good intro into it. Okay, so hang with me. We'll do like 10, 15 more minutes. And then after we go for 10 for 15 more minutes, we'll kind of take a break and we'll go over and we'll work on some lab stuff. Okay, I'll do some models and stuff like that. Does that sound good to everybody? So we'll go a little bit longer and then we'll go back and work on some lab stuff. Okay, and then I think I'm probably going to do some practice questions too, because I feel like that would really be a good idea for us, some practice questions. So we talked about the different layers, right? We said that the big vessels, like the arteries, the veins, and the arterioles and venules, we said that those all have 3 layers, but the capillary bed just has 1 layer. So if we're looking at how the capillaries actually change their shape, so when we're looking at the capillaries, we're going to see that they have the ability to like make little holes on themselves. And so there are 3 kinds of capillaries. Number 1 is called the continuous capillary. So the continuous capillary has no gaps. It is interlocked fingers. So basically the walls allow for nutrient, waste, and gas exchange. They are really great at allowing things to pass through their walls, like oxygen, carbon dioxide, and waste products. So this would be our continuous capillary. The next type of capillary is called fenestrated, the fenestrated capillary. Okay, and this guy has little tiny holes. So little holes in the wall. So little holes / small gaps allow a little blood to leak out, okay? Not blood cells, but more like little tiny particles So we're going to kind of picture little particles kind of leaking out here. So this is fluids and small solutes leave the blood. So because we have these little holes, fluids and small solutes can get out and get into the surrounding little tissues here if we have a fenestrated capillary. You need to know the locations of these fenestrated capillary. So they're in the endocrine glands, they're in the kidneys, they're in the pancreas, and the intestine. So that's the fenestrated capillary. Next, we have the sinusoid capillary. Sinusoid are going to have very large gaps. So very big gaps, large gaps, allowing blood cells to leave. So these gaps are big enough that we can allow blood cells to leave. The location is going to be the lymph nodes, the adrenal glands, the spleen, and then the bone marrow. Okay, so those are three kinds of capillaries. One has no gaps at all, called continuous. One has little holes, and that's called fenestrated. And then one has large gaps, and that's called sinusoid. Okay. Both fenestrated and sinusoid fall under the same category. I'll draw it over here. They are both an example of a discontinuous capillary. A discontinuous capillary is a type of capillary with holes versus a continuous capillary has no holes, right? I'm going to go back really quick here, and then I'll go back to that picture in 1 second, but I'm going to show you what I mean. So continuous has no holes. There's only one type, continuous. Discontinuous has holes. And there are three types of the discontinuous. There's two types, sorry. There's the fenestrated and the sinusoid. Both of those are examples of the discontinuous. Remember, the fenestrated have the little gaps, and then the sinusoid have the big gaps. So the question on the test is, which of these is not a type of capillary? And then these are our options: continuous, discontinuous, fenestrated, sinusoid, and none of these. So which of those is not a type of capillary? Good. Good. I like that you guys are both guessing. Okay, so the answer is none of these. And that's because all of these are different types of capillary. So you would think, well, discontinuous is like a category, but in a sense, discontinuous is a type, okay? If it asks you which of these is not a category, right? Fenestrated and sinusoid are not their own category. They're just like examples. But this is a specific question from the test, not, well, the test I took a long time ago, but you could see it again. Which of these is not a type of capillary? And we would say all of those are types. Right? So looking back again, continuous capillary is a type, fenestrated capillary is a type, sinusoid capillary is a type, and then the discontinuous capillary is also a type because it's a type of capillary that has holes. So the process of diapedesis is when we change the continuous capillary to the fenestrated. So this is called diapedesis, and you need to know about it. It's basically, again, when we change the continuous capillary to the fenestrated capillary. So we make little holes, changing continuous to fenestrated. And if you think about it, why would your body want to change the continuous capillary to a fenestrated one? Well, maybe it wants to make little gaps to allow things to escape, right? So this is going to allow fluid to escape, specifically white blood cells. So this is used for fighting infection. And the white blood cell, we remember, that is responsible for performing diapedesis is basophils. So basophils release histamine, and histamine causes diapedesis. Basophils release histamine, histamine causes diapedesis. And we're going to talk more on Friday about all the different functions of the white blood cells. But for now, diapedesis is caused by basophils that released histamine, and it's the changing of a continuous capillary to a fenestrated one. All right. Any questions at all? Okay, so let's, like I said, let's talk a tiny bit about some of the functions of blood and a little bit about the intro into blood, and then we'll kind of cut it off and we'll resume on Friday with more about blood, and we'll go over some models, and we'll do some practice questions as well. Probably practice questions first and then models. So looking at our little outline here, we talked about the function of capillaries, right? We talked about the 3 layers of the anatomy, the tunicas, interna, externa, and media, right? And we talk, we're going to talk a little bit about the anatomy of blood, the plasma, and the formed elements, and then we'll leave blood clotting for next time. Like I had said, PQRST, we talked about that, and electrical connection was like the SA to the AV to the bundle of His, and etc. And PQRST we talked about. So it's kind of what we've done today. Cardiovascular is a pretty big topic, so it's going to be the majority of your test for sure. So remember, cardio is the heart. We spent a long time talking about the heart, blood flow through the heart, all of the PQRST, all the electrical conduction. Vascular is the blood vessels and the blood. What did we just talk about? We just talked about the blood vessels, right? Over here, blood vessels, and we talked about the blood. We're talking about the blood now. So vascular is both the blood vessels and the blood. Cardio is everything we talked about before. So for the blood, this is what's flowing inside of the blood vessels, right? What are its functions? It's going to transport oxygen, waste, nutrients, and water. It also has like blood cells, right? These are, remember, the red blood cells are the ones that are primarily transporting the oxygen. It's going to also function for regulation. It helps to regulate temperature via vasoconstriction and vasodilation. So what's going to happen is vasoconstriction will occur at the surface if you're cold. So if you're cold, it's cold outside, your body is not going to want to make the blood cold, and it's vasoconstricting at the surface, and it's going to vasodilate to the essential organs to keep you warm. And this is if you're cold. If you're hot, it's going to vasoconstrict to the organs, and then it's going to vasodilate at your skin. You're going to be busy sweating and trying to cool down, and we're going to vasodilate at the skin if you're hot, and vasoconstrict at the organs if you're hot. If you're cold, it's going to be the opposite. Vasoconstrict at the surface of your skin and vasodilate to your organs. Okay. It also regulates blood pressure, also through vasoconstriction and vasodilation. So it's going to vasoconstrict certain areas, vasodilate others, and it regulates the oxygen content in your blood, helping you with how you breathe, basically, by keeping track of that. It also regulates hormones, and it also helps with the regulation of your acid-base balance. Another function of blood is protection. The blood has your white blood cells, and it has the ability to clot to keep you from bleeding out, right? That's a way to protect you. Your blood also performs storage, right? So it stores like oxygen, waste. This is kind of the same stuff as transport, nutrients. Remember that hemopoiesis, right? The making of blood. We learned about that last week. That was hemopoiesis. Hopefully your wheels are turning, trying to remember, where was that made? Was that made in the spongy bone or the compact bone? It's the spongy bone, right? What are the other names for it? Myeloid tissue or red bone marrow. Remember, that was made where there are no osteons. It uses the trabeculae, right? All that stuff sounding familiar. There's actually two types of hemopoiesis. There's erythropoiesis. This is for, I'll just write out RBC. Is that good with everybody? Red blood cells, RBC. And then there's leukopoiesis. And leukopoiesis is the creation of white blood cells. And this actually happens in the lymph nodes. So leukopoiesis, white blood cells, in the lymph nodes. Technically, they mature in the lymph nodes and they're created in the myeloid tissue. So again, all blood is created in the bone marrow, but the leukocytes, or the white blood cells, mature and grow up and get like fully functioning in the lymph nodes. So they're not made in the lymph nodes, they just get to full functioning maturity in okay, so that's the making of blood and then the functions of blood. Any questions at all there? Okay, so let's talk a little bit more about what the blood is made of, and then we'll kind of go towards doing questions on it. So the blood is one of our connective tissues, right? So remember that that means that it has cells and then it has matrix. There are 4 liters of blood, cells and matrix. The cells we call the formed elements, and then the matrix we call the plasma. Plasma, formed elements. The formed elements are going to be broken down into red blood cells, white blood cells, and platelets. The red blood cells we call hematocrit. The white blood cells and platelets are called buffy coat. And altogether, those are called the formed elements, making up 45% of blood. 55% of blood is made of or is the plasma. So if we picture blood kind of in like a little container, 45% is the formed elements. Like I said, red blood cells, white blood cells, and platelets. And then 55% is that plasma. And the plasma breaks down into, oops, my little guy over here. Plasma, 92% of plasma is water. So we look at that plasma by itself, 92% is water. We'll kind of draw that in blue here. The remaining 7% are proteins, and then obviously 1% more is like other substances. You don't really need to worry about that. We're concerned with the 7% proteins. So what is going on with the 7% proteins? So there are actually 4 different types of proteins you guys need to know. So the first main one is albumin. Albumin is definitely the most important. It is the most common protein in the blood. We look at that in a lot of our patients. We look at their albumin levels. Somebody has low albumin, they maybe are not eating enough protein, maybe they're in liver failure. So that's definitely something you want to remember from this class, is that albumin is the most common protein. Globulin is the second most common protein, and it's used to make our hemoglobin. So that's really important. Globulin is used to make hemoglobin. So this is protein number 1, protein number 2, and then we have 2 more proteins. I'm going to draw them here because I'm kind of running out of room. So we have fibrinogen, okay, and we have prothrombin. Both fibrinogen and prothrombin are used for clotting, and I'm going to talk about those on Friday. So these are what we call clotting factors. They're clotting factors because they help with clotting. So albumin and globulin are not clotting factors, just fibrinogen and prothrombin. If you talk about plasma without the clotting factors, what would you have left? If you have plasma and we take out number 3 and 4, what do we have left? We have water, albumin, and globulin. That is called serum. So on your test, serum would be plasma without the clotting factors. What would be left? Albumin, globulin, and water. Does that make sense? All right, things that could be on your test. Hematocrit, Buffy coat, asking you to know what is hematocrit, the red blood cell portion of blood, right? Buffy coat, the white blood cell and platelets portion. What is formed elements? The cells in blood, making up 45%. What is plasma? The water and proteins in blood, making up 55%, 92% of that is water, 7% is protein, and then knowing the 4 different proteins, which 2 are the clotting factors, and then which one, what the definition of serum is. All right, so I'm going to pull up some questions. Anything that we need to go back to? This is blood. We talked about the components and the anatomy of blood, and we talked about the heart a little earlier today. Okay. Any questions at all from anyone? Okay. I'm going to go ahead and hop in, and we'll get going on some practice questions. I feel like that would be helpful. If anyone thinks of questions as we're going along, just let me know. All right, everybody see this okay? So I'm gonna, do we want to do joints? Maybe let's skip forward and do some stuff on cardiovascular. And if I don't have enough questions, I'll stop and we'll take like a 5-minute break maybe, and I'll make some more questions, okay? So let's start with question 25, and we'll kind of go through some of the stuff with the heart. And like I said, I may do a 5-minute break and then make some more questions in just a minute here. All right. So let's start with question number 25. What is a buildup of fluid in the space around the organs? What is that called? So which of those is not true?
They're answering in the chat box.
Yes, that would be great. Good. So you guys remember that when we learned about these different buildup of fluids, right? We learned that pericarditis is the buildup of fluid around the heart. Pleuritis is actually an infection, right? Peritonitis is a buildup of fluid around the organs. And actually, when you learned about it, pleurisy is actually true of the buildup of fluid around the lungs. So the correct answer here is actually B, because pleuritis represents an infection, not a buildup of fluid. Whereas pleurisy does represent the buildup of fluid around the lungs. So that's kind of a tricky question because it's making sure that you guys have an understanding of what are the correct names, and pleurisy is technically the name for the buildup of fluid around the lungs. Okay. That was a little tricky one. So let's answer 26. The lub sound originates from which? Good. So that's the AV valves closing, right? So when our lub sound of our heart closes, lub sound, we hear the AV valves closing. One of the ways I just kind of thought of to remember it, I don't know if this will work for anybody, but lub kind of sounds like the word lab. And lab, we could think of like A, like AV. I don't know, that might work for you. Dub sound, remember, is going to be our semi-lunar valves closing. Okay, I don't really have a way to remember that, but I can think that with the lub kind of sounds like lab. So that would be, the correct answer would be B, like everybody was saying. Good. All right, which of these is true of the pulmonary arteries? So let's answer question 27. Which of these is true of the pulmonary arteries? Do they bring blood back to the heart? Do they bring blood to the systemic capillary bed, to the systemic cells, to the bronchioles, or none of these? Good. So I have a little bit of a variety of answers here. So the pulmonary, everybody is on the right track with your reasoning, so that's great. So the pulmonary arteries supply the actual alveoli with blood, because they will branch into the pulmonary capillary bed to supply those alveoli. So if that's the case, they actually can't supply any other tissues with blood, because the alveoli is really where the pulmonary capillary bed is kind of going, if that makes sense. So a lot of you were answering D, which is really good because you were thinking on the right track. It's going to the lungs. But technically, the bronchioles are just airways or openings, and it's not where the pulmonary arteries go. So our answer here would be E, none, because the bronchioles are simply airways, and they aren't really the destination of where that is supposed to go. So again, another one to kind of make you think and maybe, I guess, not trick you, but make you problem solve. All right, let's go to question 28. The brachiocephalic artery branches into the right common carotid and right subclavian. Is that true or is that false? Good, so that would be true, right? So remember that when we're talking about that aortic arch, our little aortic arch starts out with the brachiocephalic trunk, which is also called the brachiocephalic artery. And then we have the left common carotid, the left subclavian, right? And that brachiocephalic trunk, again, is also called brachiocephalic artery. It has two names, and it's going to branch into the right subclavian and the right common carotid. So if you answered true, you would be correct on that question. So let's answer 29. Fenestrated capillaries are found. And then we're going to take a little 5-minute break, and I'm going to write some more questions for us to go over, okay, about the heart. So where are fenestrated capillaries found? And this one may be a little tricky because we just did this. Good, everybody's remembering, great. So kidney is our big location for fenestrated capillaries. So that's really important. Okay, so let's take a little, what time is it? Let's come back at 6:10, and I'm gonna write some questions for the heart, okay? I'm gonna try to write up some questions for the heart. Everybody take a little break, and I'll see you guys at 6:10. I'm gonna try to get us some practice questions going. in or never left and we're just doing something. So I made a few. I think I made about 10 or 15, and then we can go back and find some more. But I thought this would be kind of a good little walk through the heart. I think I made, let's see, 1, 2, 3, 4, 5, 6. I should really number these, so make it more. Take 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. All right, so when picturing the heart, it has a variety of layers. Which of these is a layer around the heart itself? And then everybody can just, you can put your chat answers in the chat. If you want, you can send them privately to me. I can still see them just so that we can all participate. Good. All right, so I have a pretty good response amount of answers here. So the myocardium is actually the one that is the layer around the heart itself. So remember that when we see that wording, the heart itself, we're talking about 3 options: the epicardium, the myocardium, or the endocardium. Those are the ones that are the layers around the heart itself. The other ones over here, fibrous, serous, and parietal, those are actually the layers around the heart. But when we say the heart itself, we're trying to show you that the question is asking for the ones that are a lot closer to the actual chambers of the heart. So it's a little bit of a tricky wording, but you're going to see it worded that way on the exam. So which of these touches the epicardium? Good. So the epicardium, right, is not directly touched by the fibrous layer. That's the one that's all the way outside, right? But we know that the next layer, we kind of picture our layers. So there's the fibrous layer, and then the next layer in, we've got that parietal layer. So I'm going to write P. And then the next one in is the visceral layer. And remember that between the parietal and the visceral, what do we have? We have that serous space with the serous fluid, and those two together are the serous membrane, right? And then the next thing we have is the epicardium, and the epicardium sticks very closely to the visceral layer. So this is the epicardium. Okay, so that's the answer here. That's the layer that sticks the closest. And then the next thing we have would be the myocardium. And then the very last layer on the inside is that endocardium, right? Okay, so remember to go over those. They will be quite a bit on the test. Okay. All right, let's do the next one. I did quite a few about the heart layers because I thought they were kind of a weak area. So which of these lines the actual heart chambers? Good. So the endocardium, that's the one that lines the actual heart chambers, right? That's the one that keeps those lined. Good. All right. So number 4, when the blood passes from the atria down to the ventricles, it does not flow through which of these valves? Good. So the aortic semilunar, right? So remember that the mitral is also called the bicuspid. This is on the left side, and it's an AV valve, which means it's atrioventricular, passing from the atria to the ventricles. And then there's the tricuspid, which is the right AV valve. And then the aortic semilunar, right, is not one that flows from the atria to the ventricles. All right, let's check out the next question. The aortic semilunar is found leaving where? Good. D, right? So we know that when we have that blood flow through the heart, that aortic semilunar is on the left side. I, again, have already said this, I think, but on your test, I recommend making a quick drawing of your heart so you don't lose track of your right and your left, because that can be really easy to do. But the left ventricle, again, would be pumping out to the body through the aorta. So you're right. So the aortic semilunar valve would be found leaving the left ventricle. All right, so question 6, the coronary arteries supply which of these? Good. The myocardial cells, right? Number B, the myocardial cells. So the coronary arteries are going to be supplying the actual heart cells. So the myocardial cells. It seemed like it was a pretty good one for everyone. Okay, so when the chordae tendineae break, it can cause which of these? Good. So everybody's saying valvular prolapse. Very good. So that's the little chordae tendineae that hold the valve. I'm going to do a little bonus question here. How many chordae tendineae are there total in the heart? Let's see. So everybody, let's answer this one right now. How many chordae tendineae are there in the heart? Okay, I don't know. I don't think I'm having any guesses. Does anybody want to guess for me? Oh, there we go. Wait. Okay, so you guys are guessing D, right? So D would be correct. I think that's from this question, unless that's from the last question. So there are 5, and this is because there are 3 for the tricuspid valve, and there are 2 for the bicuspid. Sorry, that typed as 3. There are 2 for the bicuspid valve. So if you said 5, that would be correct. So there are 5 chordae tendineae. There are also 5 papillary muscle. So if you're asked how many chordae tendineae are there for the tricuspid valve, it would be 3. How many papillary muscle for the tricuspid valve? 3. Good. So everybody, does that make sense to everybody? 1 for each cusp of the valve. one chordae tendineae, one papillary muscle for each cusp. Okay. That is a great question. So the semilunar valves do not have chordae tendineae because of their shape and basically the way that the blood pumps out of the heart. Not enough volume of blood comes back against the semilunar valve to risk prolapse. So they are not prone to prolapse because of the way that the blood moves through the vessels. And so basically just those valves don't have the tendency for prolapse, so they don't need chordae tendineae and papillary muscle. That is a very good question. Good thinking. All right. Number 8, which of these is not true of the azygous vein? Good. So remember that the azygous vein would drain to the superior vena cava, not to the inferior vena cava. So the correct answer here would be superior vena cava, not inferior vena cava. Good, very good. Okay, let's do question number 9, and then we'll go back and find some more questions over on the other page. Question number 9, the P wave. Good, good. Atria depolarization, right? When the P wave comes, the atria depolarization occurs. All right, let's do a quick fill in the blank. When does B happen? So write in the chat, when, during which part of the PQRST does B happen? Is it P, QRS, or T? Good, QRS, right? So during the QRS complex, we have atria repolarization. Ventricular depolarization is also during the QRS, and then ventricular repolarization is during the T. Very good. Okay, so let's take a look at some more questions. I'm going to go over here. Okay, so let's go up. Let me go. Don't really want to do bone and cartilage right now. Let's go over to these ones. And on Friday, I'll plan to make some more questions as well. I like taking a break, especially when it's more lecture heavy, and then writing some questions and coming back to it. Okay, so let's stick with heart since we're kind of on that right now. So 4, the fluid that pulls away heat and reduces friction is found internal to which layer? So this one's going to take a little bit of time to decipher. So what is that fluid, and then where would be the location internal to which layer? Good. Okay, so if you're somebody who put down E as your answer, can you write what you think the correct answer is? If none of these are right, what is the correct one? Good, right? So this is trying to ask you for the pericardial layer. So internal to the pericardial layer would mean closer to the heart. And this fluid that pulls away heat and reduces friction would be found closer to the heart, internal to the pericardial layer. So between the visceral layer and the pericardial layer, so external to the visceral layer, internal to the pericardial layer. So this is kind of a tricky question, but the answer there would be E, because we're looking for pericardial layer. Good job. Good job, everyone. Okay, so question 5. What motivates the tricuspid valve to close? All right, so we know that it, let's start with ones we know it definitely isn't. It's not D, right? It's not the closing of semilunar valves causing it to close. It's not the intercalated discs allowing sodium to travel quickly. That's what motivates the ventricles to contract, not what motivates the tricuspid valve to close. Same thing with Purkinje fibers traveling up the myocardium. That would be for contraction of the ventricles. And the SA node sending signals to the AV node, what did we talk about tonight? We said that that was for atria depolarization, right? So the atria depolarize when the SA sends the signal to the AV node. So what motivates the tricuspid valve to close? What motivates, oh, sorry, I don't know why this is all caps. There we go. What motivates valves to close? Pressure. So if you answered E, and you were thinking it's the pressure of blood that causes valves to close, that would be correct. So unless you see pressure motivating the valves to close on your exam, you're not going to be choosing that option, okay? So there might be ones that are there to trick you, but you will just choose pressure as the answer. So 6, what is the purpose of trabeculae in the heart? Kennedy, you asked where the answers are. I have this posted as well as the answers at the end. If you want like explanations or where you're supposed to be getting this content, all of this is coming from your lectures. So if you're taking Bender or Newton, it will be coming from your lectures is the source of all of this information. So for question 6, what is the purpose of trabeculae in the heart? They are going to help with the accommodation of blood. Very good. So when those ventricles are refilling, right, they're going to help with refilling and accommodating that refilling in the trabeculae, in the ventricles. Very good. Okay. So let's keep going here. So for number 7, when blood flows out of the left ventricle, what is the very first structure it will meet up with? Good. C, right? So it's going to leave and it's going to go to that aorta first. Good. Okay, when blood returns to the left atria, it returns through which vessels? Good, good. So blood always returns to the heart through veins. Veins come back to the heart. Arteries go away from the heart. So if it's coming back to the heart, it's always coming back to the heart as a vein. Good? Great. Good job, guys. Okay.
Quick
So number
question.
9. Yeah.
I'm sorry, my computer just died and I missed what I wrote. But these answers that these questions and answers, where like I'm having trouble where I should focus my studying. Should I focus it on the textbook? Is that where most of the questions from the test would be, or would it be in the lectures itself?
Everything for your test are found in the lectures themselves. You don't need to use the textbook at all. It's kind of like a supplemental if you so desire and you learn well from textbooks, you could open it up. But when I took the class, I didn't use the textbook at all. And so you can just study the lectures. I just took really detailed notes on the lectures. And then I went over those and reviewed those a lot every week, kind of every day. And then I would review, especially on the weekends with like a friend or something. So the lectures is where you want to focus your time, but don't just print out the slides and then take a few notes and think that you got enough. You really have to like pause the recording and write down everything the professor's saying and replay it and pause it again, because a lot is being said that's not necessarily on the PowerPoint slide or maybe not written on the board, but you still need to know.
Okay.
Does that help?
Thank you. Yeah.
The blood that enters the right ventricle originated from where? Good. So all of these, right? The superior vena cava, azygous vein, coronary sinus, and inferior vena cava. All of those dump blood into the right atria, which will then flow into the right ventricle. Question 10, when blood circulates to peripheral tissues, which chamber of the heart is that blood coming from? Good. D, right? So the left ventricle pumps blood out to the body. Very good. All right, you guys ready to do some on joints? That's what we talked about on Thursday, right? So let's get your kind of wheels turning back to joints. I think that some review would be good. So which of these is not true of gomphosis? The gomphosis joint. You remember what gomphosis was? So which is not true? Is it linked to the gums? Is it synarthritic? Right. So when we talked about the joints, we said that there basically are 3 types. There's fibrous, which are made of dense regular, and those have our gomphosis, which are found in the gums, the suture, and then the syndesmosis or interosseous, and that's between the radius and ulna and tibia and fibula. Suture are between the skull bones. And then our next one is our cartilaginous, and that was between cartilaginous, that is our synchondrosis and our symphysis, and then lastly, synovial. So the gomphosis are linked to the gums in the mandible. That would be true. They are synarthritic. You don't want your gums to move. So C would not be true because all fibrous joints are made out of dense, regular connective tissue. Okay. So review some of your, don't forget to review your articulations content. This is good stuff that you can go over and kind of spend some more time reviewing. Let's do a few more of these questions here on the heart. So for question number 13, a patient with a partial prolapse has lost function to which heart structure? So remember, what causes prolapse? So what causes prolapse? The breaking of the chordae tendineae. So our correct answer would actually be E. Papillary muscle function to hold the valve to the floor of the ventricle, but they don't prevent prolapse in any way. The definition of the structure that prevents prolapse is the chordae tendineae. Chordae tendineae prevent prolapse. Quite a few people answered trabeculae. Trabeculae increase surface area basically, but they do not function to prevent prolapse. So our answer would be E. The dub sound of the heartbeat is caused by what? Good. The semilunar valves closing off, right? The AV, the lub sound, you could think like lab AV valves, dub sound, semilunar valves. All right, 15. Blood leaving the heart and going to the lungs will go into the pulmonary trunk, which branches into what? Good. Pulmonary arteries, right? So the blood is going into that pulmonary trunk, and then it's going to branch into the pulmonary arteries. Good job. Good. Okay, so the pulmonary circuit ends in which chamber? Question 16. Good, nice. So it starts in that right ventricle and it ends in that left atria. Very nice. Good job, guys. Great. Okay, so let's do 17. And we'll do, I think it looks like 18 and 19. And then we'll kind of see where you guys want to go from there. 17. Infants do not oxygenate their own blood. Which heart structure is essential to their mother oxygenating blood for them? Good, the ductus arteriosus, right? The ligamentum arteriosum, the function of the ligamentum arteriosum, which is what it becomes when it's an adult, is to hold the aortic arch in place. Whereas the ductus arteriosus, like you said, provides patency between the pulmonary trunk and aorta in babies. Okay, great, good job. All right, that's one you could be asked on your lab exam, so make sure you know that. It's one that could appear on your lab exam. All right, let's take a look at these last few here, 18, 19, and 20, and then we'll just do some question time. So the tunica media is not found in which of these vessels? B, right? We said that the only layer found in the capillaries is that tunica interna, right? The tunica media, tunica externa is not found in the capillaries. Very, very good. All right, so 19, the process of transitioning a continuous capillary to a fenestrated one is called what? What do we call that process? Good. So B, diapedesis, which I talked about. It's that process of converting a continuous capillary to a fenestrated one. All right, 20. When blood returns to the heart, it uses muscle movement along with which of these to help move it? Good. D, right? Valves. Valves bring blood back to the heart. All right, so I feel like that's a pretty good coverage of our cardiovascular content. What I was wanting to do was see if we had any models to go over. And then I'm gonna see, I think that we're kind of caught up on models for right now because we just did the heart model last time. And the next one would be the axial, which I haven't gone over respiratory yet, so I'm not going to cover that. For bones, this unit, I usually don't cover bones. If you have any questions on something you're having a hard time finding, let me know. The cardiovascular lab I talked with you guys about on, I don't know, Thursday or Friday, I think it was. Does anybody have any questions at all about the cardiovascular lab? I think somebody had written me about the blood flow. Now that you guys have a really good understanding of the blood flow from kind of like all the questions we did, I was just seeing that everybody was doing a really good job. I'll just write in the answers for that, so that saves you a little bit of time and you can see if you got it right. Remember to use these last 4 pages to fill out the last 2 pages. So remember that on the very end of this packet, you guys will both, Bender and Knudsen, be having a quiz on this content on the last 2 pages. And what you're gonna do is you're gonna use the Google Drive link with these pictures to fill that out. If you were in Bender's class, I had somebody write me saying they couldn't find the link. If you guys go to the actual assignment for the cardiovascular packet, if you're in Bender's class, you go to that assignment. The Google Drive link is found in the cardiovascular lab packet assignment. If you're in Knutson's class, it would be in the module. So if you guys click this cardiovascular homework assignment, right here, that Google Drive link is the same Google Drive link I'm on right now, okay? So let me just fill out that little part here. Oh, that looks a little weird. Let's go over here. Again, use those diagrams to fill those last 2 pages out, and you can study that. Okay, so that's for your quiz that's coming up. Okay, so the blood leaves the left ventricle, and you guys answered it over and over tonight. Where's that first place it goes? The aortic semilunar valve. From the aortic semilunar valve, it goes to the aorta. The aorta will then turn into arteries, systemic arteries, then into systemic arterioles, a systemic capillary bed, systemic venules. This is what I was going over tonight. And then systemic veins. The inferior and superior vena cava is what those merge into. And where do the inferior and superior vena cava go? Everybody again was saying this tonight. They go into the right atrium. Good? Does that part of that makes sense to everybody? Okay, so I'm going to erase it. So if you want to take a picture, that would be the time. Okay. All right, so from the right atrium, where does it go? How does it pass through to the ventricle? From the right atrium, it goes through the tricuspid or right atrioventricular valve. There is a typo in the packet and it says left AV valve tricuspid. That's not right. It's tricuspid right AV valve. Then it goes to the right ventricle. I don't know how that typo ended up there. From the right ventricle, it's going to leave and it's going to go through the pulmonary semilunar valve. From the pulmonary semilunar valve, it's going to get into the pulmonary trunk. From there, the pulmonary arteries, the pulmonary arterioles, the pulmonary capillary bed, the pulmonary venules, pulmonary veins, and where do the pulmonary, oh, I just realized this wants me to, I thought that this was one thing, but it's actually not. It looks like it's 2. It wants me to do it like this, capillary bed, and then each of these, because I need room for my other valve. Pulmonary capillary bed, pulmonary venules, pulmonary veins, left atria, right? That's where the pulmonary veins empty into. And then the left atria is connected to the left ventricle through the bicuspid, mitral, or left AV valve. Okay, so if you wanted a picture of that, you can get that now. All right, are there any other, does anybody have any other questions about the cardiovascular packet?
Yeah, I have a quick question
Yeah.
on the 5K. I couldn't really understand 5K.
Mm-hmm.
It with the SA node list in order. I had trouble with finding the list.
Got it. Were you here for the beginning of the SI today at all, or were you only able to come a little later?
I was here.
Okay, so you should be able to kind of go through that based on what we talked about today, right? So the SA node, what does that go to next? The AV node.
The AV. Oh.
Yeah. And then the bundle of His would be next. And then the interseptal fibers. And then remember how I said that you have the Purkinje, and then one Purkinje fiber goes back up to the SA to start it over. So then that would be, and then it would just be kind of a revolving circle. Any other questions? Right, yes, the right palmar metacarpal would go to their own digital arteries. Yes, because the palmar metacarpals are just basically on your fingers, on your palms of your hands, and then they would go to the digital arteries on your fingers. So yeah, they would split and go. All right, any other questions at all?
One quick question again, I'm sorry.
Yeah, no problem.
So just want to make sure after the inferior vena cava, it goes to superior vena cava.
So the inferior vena cava and superior vena cava both drop into the heart, into the right atria together. So that's why it says and. Does that make sense?
Okay.
That they kind
Yes.
of join together. Someone was saying, you're saying a mistake with the median cubital. Do you mean how it's like underlined right here? That's not a mistake. I think that whoever it was just underlined it. This is still correct. There's nothing wrong with that one. Does that answer your question? Okay, so if you guys have any other questions for me, we have, oh, I was supposed to end at 6:45. I'm so sorry. I thought that it was 7 and I don't even know. It's already overtime. Anyway, sorry, I was thinking 7. I will see you guys on Friday morning, 9 to 11. Thank you guys so much for coming tonight.
Thank you.
Bye.